microRNAs, and small nuclear RNAs. So we so obsess about genes that encode mRNAs that encode proteins. Those are the only things that we really call genes, but it's not right. Genes encode all of these, but some of these genes encode final RNA products. We probably won't mention Pol1 or Pol3 again. From here on out, we're going to really focus on Pol2. Pol2, like RNA polymerase in prokaryotes, does not find the promoter at all. It can't see it. What finds the promoter is something called the TATA binding protein, which will bind to the TATA box, which is AT-rich. But that is the name of that particular segment that defines a promoter in a eukaryotic gene. TATA binding protein comes to this site together with something called transcription factor for RNA polymerase II subunit D, so TF2D. Then TF2B jumps on, then TF2E, TF2H, TF2F plus RNA polymerase II, and so they all kind of gather at this site and get RNA polymerase II positioned correctly so that the double helix can be opened up just a little downstream from the TATA box. Transcription begins, the carboxy terminus gets phosphorylated, and all of this stuff that got RNA polymerase in position has to let go, just like sigma had to let go, so that the RNA polymerase II can now take on the high mobility configuration and go coursing down the chromosome, transcribing that particular gene unit. So we've seen this theme before. In eukaryotes, many more participants, all subdividing the work. Sigma did this whole job, but now we need TF2, A, B, C, D, E, F, G, H to get all that done. So that gang are sometimes referred to as the basal transcription factors. And I like to reserve the term transcription factor to mean just this set of basal transcription factors. At the beginning of this field, I don't know, in the 1980s, anything that affected transcription was called the transcription factor. I think it's gotten too confusing. A bunch of books now are reserving the term transcription factor, or TF, to mean just that set, the TF2 A through H. The basal transcription factors required to find the TATA box and get RNA polymerase II in position. They're not very interesting because they seem to be required constantly. They're almost like a housekeeping gene is what we call it, right? It always has to be there. But the theme is different vocabulary than in the prokaryotes, more participants, and we don't want to mix any of that up. This is what the TATA binding protein looks like. Again, just to keep you thinking about shapes, we've got the alpha helix, we've got a little bit of beta sheet, we've got a quaternary structure as these two are interacting. They actually touch this particular sequence of bases. They actually bend the DNA, and that bent DNA, you can see how that's going to be a little easier to kind of pry apart. So these days, it's not enough just to know the consensus sequence, blah, 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 but you kind of want a three-dimensional diagram of exactly what's happening, maybe four-dimensional, because things change through time. And this is the sort of detail that we're starting to see on a kind of daily basis. Now, the more we study the initiation of transcription in eukaryotes, the more and more complicated it gets. A lot more is involved than just the basal or general transcription factors. In addition, if you had just this, you could get RNA polymerase II to be brought to the right place, but it might just as soon get up and leave. And so there's other stuff involved. There's these other proteins, shown here and here and here and here, that seem to be required to activate transcription. Basal factors get everything lined up and ready to go, but what makes it actually go is this other stuff that seems to be binding to sequences in the DNA, sometimes a little bit upstream of Tata, but sometimes incredibly far upstream from Tata. Sometimes I think stuff from way down the street could come back like a kite on a string and have an effect over here. I'm even starting to worry, and maybe I'm the only one, that why wouldn't a protein stuck to a different chromosome be able to come on over here and activate the transcription of this gene? So there's a lot going on in three-dimensional space that we don't quite understand. One of the things we're imagining is these things bind to elements. So these would be elements in DNA, particular sequences in the DNA to which these particular proteins bind. Rather than call any of those things transcription factors, our book and several others are now calling these things transcription activators. The transcription factors must be there. The transcription activators, that's where you can sometimes have them available and sometimes not. Now, that's not the whole story yet. Now people are describing proteins that also stick to promoter elements in the DNA, but instead of activating transcription, they might be acting to silence transcription. And what we're starting to now hypothesize is all of these different proteins are not contacting and tickling RNA polymerase. They seem to be talking to a middleman who is, like in Iowa, accepting all of these votes. Some of the votes say, yes, transcribe, and some of the factors instead say, no, don't transcribe. And there seems to be some signal in both directions. The mediator is the protein that is actually touching this consortium of activators and silencers and deciding whether or not it's going to phosphorylate the carboxy terminus, which we said a slide ago is what makes the RNA polymerase II take off down the street. What a hopeless mess. But that's why we're gorgeous and wonderful and not boring like bacteria, which are perfect. Bacteria are perfect. Don't tell Michael I insulted them. It's just, this is just simply hard to understand, but it gives us all this plasticity. So it's a wonderful thing that we're this complicated. Did you have a question?
Oh, I was just gonna say, I took a class with Dr. Mervani and
Yeah.
Activators and things like that can come from other chromosomes.
Yeah,
to activate.
I haven't read a paper about it, but I am happy to hear.
Yeah, it does happen.
Yeah, so we have to stop thinking about DNA as a line, which we thought of 100 years, it's a line. It's in a nucleus, there's three-dimensional space. I mean, just because chromosome 2 hangs out over here, in some cells, chromosome 5 is going to be like touching it. They got to be cross-talking. And we're not set up that way. If you're linked, maybe it's important, but if you're unlinked, independent assortment, Mendel said they're completely had nothing to do with each other. I don't buy it for a minute. How we're going to figure this out is going to be difficult, but that's why you're young and strong and full of imagination. Might be icy in the morning. That won't affect us. I'm not going to see you till Friday, right? It'll be all better by Friday.